Welcome back to my YouTube channel. Today we're going to be talking about AP Bio Unit 6, which is gene expression and regulation. So in today's video, we will be talking about topics 6.1, 6.3, 6.4, 6.5, and 6.7. Okay, so starting off with 6.1, DNA and RNA structure. So DNA is a polymer of nucleotides, and DNA is a double-stranded molecule, while RNA is a single-stranded molecule made up of nucleotides that contain the sugar ribose. Both strands are made up of a backbone that's composed of alternating sugar phosphate molecules. And then the two strands that make up the DNA double helix are anti-parallel, so that means they're just going in opposite directions. One strand runs in the 5' to 3' direction, while the other one runs in the 3' to 5' direction. So the two strands twist around each other, forming a double helix. That's how DNA is formed, and they're held together by hydrogen bonds between nitrogenous bases. So a nucleotide is the monomer of DNA. It contains a nitrogenous base, a 5-carbon sugar, and a phosphate group. Pentose sugar is found in DNA nucleotides and is deoxyribose. The carbon atoms in the pentose sugar are numbered 1' to 5', and then the nitrogenous base binds to the 1' carbon, and the phosphate binds to the 5' carbon. So with nitrogenous bases, there are two types. There are pyrimidines and purines. So pyrimidines are bases with one ring and include cytosine and thymine and uracil, whereas purines are bases with two rings and include adenine and guanine. So a way to remember purines is through the phase pure as gold, which indicates like purines are adenine and guanine. In DNA, there are only cytosine, thymine, adenine, guanine, whereas in RNA, there's no thymine, there's uracil that replaces it. And with base pairing rules, a purine always binds with a pyrimidine, so like guanine with cytosine. And then there's DNA in prokaryotes and DNA in eukaryotes. So the DNA inside prokaryotes are organized in a single chromosome, but in eukaryotes they're organized into linear and plural chromosomes. In bacterial cells, viruses, and some eukaryotes contain plasmids, and then plasmids are small circular double-stranded DNA molecules. And I think that's it for this topic, so we're going to jump to topic 6.3, which is transcription and RNA processing. So genetic info is usually stored in DNA molecules, and RNA molecules are used to facilitate protein synthesis using DNA information. So that means that ribosomes contain RNA and they assemble proteins. Information in genes flow from it uses the pattern of central dogma, so it goes from DNA to RNA, and then it goes to the protein, and that's the central dogma basically. And then information in genes flow first from DNA to RNA through transcription, and then RNA to proteins, which is through translation. And the directional flow of genetic information, as I mentioned, is the central dogma. So transcription. So the goal of transcription is to make a RNA copy of a gene's DNA sequence. So the process of transcription is when like DNA strands are separated, and then one strand is the template strand, so that's the non-coding strand, while the other becomes the non-template strand or the coding strand. The strand that is serving as the template strand depends on the gene that is being transcribed and the gene that is targeted for transcription, which, that's it. And then, so RNA synthesis can So what happens is RNA synthesis is catalyzed by the enzyme, and then RNA polymerase, which unwinds the DNA strands and joins together RNA nucleotides. So the enzyme RNA polymerase synthesizes the messenger RNA, so mRNA, in the 5' to 3' direction by reading the template strand in a 3' to 5' direction, and then that gets us to the mRNA transcript modification, which occurs in eukaryotes. So in eukaryotes, the mRNA made directly through transcription is known as pre-mRNA or primary transcript, and many modifications can occur. So one of the examples is a poly A tail, which basically it's 100 to 200 adenine nucleotides are added to 3' end, and this can be beneficial because it increases stability and helps with exporting from the nucleus. Well, on the left you can see at the 5' end there's a G2B cap, which is when a modified guanine nucleotide is added to the 5' end, and it helps the transcript and helps ribosomes attach to the mRNA. And then, so most eukaryotic genes and their RNA transcripts have long non-coding stretches of nucleotides called introns that lie between the coding regions. So the problem is that introns do not code for amino acids, but they lie like in the middle of the coding region. So to get rid of them, they have to be spliced out. And since the exons, they're the coding regions, they have to be express which they're the ones that usually translate into amino sequences. So as you can see with this photo, RNA splicing is it's just basically cutting out introns using spliceosomes, and then that leaves the exons. And then this picture visualizes it. So then there's alternative RNA splicing, which is basically when the introns are cut out, and then the splicing retention of exons are used to make like different versions of the resulting mRNA molecule. So the photo basically shows like it starts with the same version, but then there can be different ways that it gets translated. So as you can see with the photos, it can be something like protein A, B, or C because they're alternative ways to be expressed. And then our next topic is topic 6.4, translation. So translation has 3 steps. There's step 1, initiation, step 2, elongation, and step 3, termination. Also, translation occurs on ribosomes, and in prokaryotic organism, translation occurs while the mRNA is being transcribed, so it's like happening simultaneously. So initiation, that's basically just when the process starts, and our RNA in the ribosome interacts with the mRNA at the first start codon, so when the amino acid reads MET. And then I'm going to discuss tRNA before I actually get into elongation. So tRNA is a molecule that brings the correct amino acid to the correct place specified by the codon on the mRNA. And basically tRNA uses anticodons, which are complements of mRNA codons. And as I'll discuss it in a minute, tRNA helps with the elongation stage. So step 2. The newly arrived tRNA brings amino acids to a growing polypeptide chain, and rRNA adds those amino acids as tRNA brings them over. And amino acids are added one by one to the previous amino acid on the growing chain. And I already mentioned the anticodon. The rRNA molecule catalyzes the peptide bond between the new amino acid in the site A and the end of the growing polypeptide in site P. The ribosome then moves the tRNA into the A to P, and then from P to E, and during the E site it exits. And then step 3 is termination, which is basically the end. So elongation continues until a stop codon in the mRNA reaches the A site, and then a release factor binds directly to the stop codon in site A. The release factor causes the addition of a water molecule instead of an amino acid to the polypeptide, and then that causes it to hydrolyze, and the bond, which hydrolyzes the bond, which causes the release of the polypeptide, and the process finally ends. Okay, next we have topic 6.5, which is regulation of gene expression. So gene expression is the process by which instructions in the DNA are transcribed and translated into a functional protein. And then regulatory sequences are stretches of DNA that promote or inhibit protein synthesis. So some other key terms are operons, which are closely linked genes that produce a single mRNA molecule during transcription. And then we have regulatory genes, which are genes that are upstream of the operon that code for regulatory proteins. So we have more key terms that we have are that operons are inducible or repressible, and the inducible operons are involved in catabolic pathways, which means that they break down, and repressible are involved in anabolic pathways, that means they build up. So we have two examples, which are lac operons, which are inducible, whereas trip operons are repressible. So with the lac operons, since it's inducible, that means that its transcription is off and it can only be turned on by a small molecule that binds to its regulatory protein. And then lac operon is made by a regulatory gene, which is located on the outside of the operon, and when it is made in its active form, it can bind to the operator. To turn off the lac repressor, a allolactose acts as an inducer and activates the repressor protein and turns transcription back on. And then the trip operon is repressible because transcription is on, but it can be inhibited by a molecule that binds to this regulatory protein. Trip repressor is the product of a regulatory gene that is located away from the operon. So, I forgot to mention that small amounts of this repressor is always present in the cell, and the trip repressor is made in its inactive form, meaning that it cannot bind to the operator. Also, tryptophan acts as a co-repressor for this because it binds at an allosteric site, which leads to the transcription to turn off. And I think I forgot to mention beforehand that operons are composed of a promoter, operator, and a group of genes. And I believe that's it for this section. And then let's go to topic 6.7, which is mutations. So mutations are changes in the nucleotide sequence of DNA or mRNA that code for a protein. So a few benefits or just general information about mutations is that mutations are the primary source of genetic variations, and they're caused by mutagens, which are basically environmental factors like radiation, chemical exposure, or sunlight. And the environment determines whether the mutation is harmful, beneficial, or neutral. It truly just depends on the selection of the trait that's there, whether there's an advantage or if it's favorable. Mutations can be harmful if they result in the loss of a function of genes or other DNA information, and those harmful mutations can actually be passed on to offsprings, and some mutations can also lead to cancer. Mutations can also be silent, which I will discuss as they go on, and they're not as bad. They can be neutral, positive, and some can be harmful, but they're not as harmful. So types of mutations. The first one we have is point mutation, which alters a single nucleotide to another incorrect nucleotide. And an example of this would be sickle cell anemia, where one nucleotide is changed. I believe it's the 6th one, but don't quote me on that. And let's see, we have other substitution mutations. And for this one, we have a silent mutation, which has no effect on the amino acid produced by a codon because of the redundancy in the genetic code, which basically means that if you look at the amino acid chart, you can see like in the top left corner, you see UUU and then UUC. If you're to change the 3rd nucleotide and replace that U with a C, it wouldn't have an effect because it produces the same amino acid. So that means there will be absolutely no effect on the person, the gene, or anything. And another substitution mutation we have is missense mutation, which still codes for an amino acid but not the correct one. So that means just like in our last example, it had the same amino acid, and this one, if we use another letter at the end, then we wouldn't get the same amino acid. And then this can be very dramatic because if the chemical properties of the original and the new amino acid are very different, it can like lead to something dangerous. And then we have nonsense mutation, which changes an amino acid codon into a stop codon, and this nearly always leads to a non-functional protein. Okay, so types of insertions and deletions. So an insertion or deletion is just an addition or loss of a nucleotide pair in a gene, and this has a worse effect on the protein than substitutions, because substitution just like swaps something out, but this can like completely alter it. And we have, let's see, a frameshift mutation, which, as I mentioned, is an insertion or deletion of nucleotides. And what it does is it takes a nucleotide and then, as I said, it takes out or adds one in, and that leads to a downstream of, it affects downstream coding because it can improperly group codons, and that's more severe because it can just alter all the codons that are being read, and that means that codons aren't being read correctly, which leads to a harmful impact on them. And that's it.